Senescence is a really interesting process. It's actually what happens when your cells age. And so, unfortunately, as we get older, our cells will senesce. The interesting thing is that also happens in the early events in cancer. So really trying to understand how cancer cells fuel themselves and how they use these sort of normal metabolic pathways of using energy to thrive in an environment that they may not be able to thrive in if they were normal cells. My name is Katherine, and I'm interested in ovarian cancer. I think what we're doing may be relevant in a number of ways for patients. One of these pathways that I'm really interested in, like we mentioned senescence, it's an early event in cancer. And so my sort of career goal has always been, could we prevent cancer from even happening? Unfortunately, ovarian cancer patients do not respond very well to the therapies that they use in the clinic. And so some of these pathways that we're interested in, we might be able to find novel targets and maybe be able to come up with some inhibitors. And one of the great things about being here at Penn State Hershey is that the hospital is right next door. We may even be able to do a clinical trial. And that's something that actually isn't that long term, maybe in the 5 to 10-year range. It wasn't purposeful to arrange an all-female team. I have a soft spot for women and women in science because we are a minority still. And I am an advocate for them and a strong supporter of them. And so I think people who want something like that in their life maybe gravitate towards me in my lab. I'm hoping to get a diverse group of people, both males and females, and also different backgrounds and ethnicities, because that is so important for science. It's actually one of the reasons I got into science, is because I love the diversity of it. And diversity of thought makes us all better.
I'm John ELFAR. I'm the Vice Chairman of Orthopedic Surgery and Rehabilitation at the Pennsylvania State University. Nerves are what control everything, what you feel and what you can do, what you can move. They're all under nerve control. The way your brain gets those signals to your fingers and your toes and your legs is through nerves. I'm an orthopedic surgeon, so I see traumatized patients. I see patients who've had their nerves cut by the bullet that's going through the tissue, or a car accident, a bad mangling injury. These are the kinds of injuries I see. But all over the medical center, you will find peripheral nerve injuries that are the consequence of care. There's a lot of very sad patients who have the cancer out but can't move the limb, or who get treated with radiation but can't feel. 4-aminopyridine was a drug we just happened to come across. It's called 4AP, and we were using it in experiments in the lab for a totally different purpose. We were trying to use it to tell whether or not our nerve injuries were bad enough. That's currently impossible right now. Even with all of the greatest tools we have, all the greatest imaging, we can't tell whether a nerve has been cut or not. The cool thing about 4AP is it's currently used in humans. So it's FDA approved, so we have dosages that are safe in humans, for example. When we use a small dose of 4AP, we can take a nerve that looks like it's been cut and make it function for the short enough time we need to tell that it hasn't been cut. The second discovery was, if we took nerves that have been injured, we could very substantially increase the rate at which they recover with continuous dosing of this drug. 4AP has applications so far beyond my own field that I haven't had any problems interesting surgeons and physicians from other departments in helping. We have trials currently planned, and we're writing grants for trials in neurology, in neurosurgery, in urology, in general surgery, that you wouldn't expect 4AP, but yeah, actually 4AP has tremendous traction in fields where nerve injuries are an obligate consequence of care that is really important. You find other departments that are very interested. Right now, we have an opportunity, because we're on this cusp, we have an opportunity to actually try a treatment, as opposed to do the extra stuff required to justify trying the treatment. we want to go and take the test. All we want to do is take the actual test.
I couldn't be more happy and more proud of being a member of the Penn State community. My name is David DeGraff, and I'm an assistant professor in the departments of pathology, surgery, and biochemistry and molecular biology. Getting a grant from the American Cancer Society definitely opens up doors for additional funding. I think it's a true mark of distinction. I think that, number one, it helps us grow the research program and get the results that we need and really get the results that are showing us that we're making an impact. I mean, I think our donors are members of our team because the faith they have put in us and the mission that we have here at the Penn State College of Medicine is extraordinary, and we couldn't do it without them.
The goal really at the end of this project is to see that if we manipulate the expression or target the expression of these transcription factors, can we convert one subtype of bladder cancer into another subtype of bladder cancer that is more responsive to treatment?
One of the things that I love about being a researcher is it really gives me an opportunity to be an explorer. On an ideal day, I get to see something for the first time that no one else has seen. The discovery is what keeps us going, helps us deal with all the frustrating times, and that's why we do the job. So I would also thank the donors for their vision and their belief in us. They might not be the ones that are able to go into the laboratory and do the experiment, but they know that our ideas are important and they have impact, and the donors have the vision to see that. Thank you.
You are helping us
Change lives.